Biotechnology has been a great tool when it comes to the advancement of human development and different industries which concerns it. And when it comes to agriculture, biotechnology enhances the efficiency of animal and as well as crop production, which results to higher yield or more bountiful harvest. Hi, I'm Christopher Ranchio, and together with my group mates, we are Group 4 of BSA1C, tasked to present to you Article 4 titled as Plastid Transformation, Advances and Challenges for its Implementation in Agricultural Crops. With its authors Quintin Rascon Cruzque, Gonzalez Barriaga CD, Iglesias Figueroa BF et al., and its place and date of publication as given Pontificia Universidad Católica de Valparaíso in the year of 2021. Proceeding now to the second slide of the presentation, two terms are defined by the article and are considered as key terms or the center of the ideas behind the research. First on the list is the term plastid transformation, which is defined by Cruz et al. In the year 2021 as the process of altering the plant cell, specifically called as plastids, which generally pertains to the whole organelles of the plant cell which contains pigments and are concerned or a part of the process of photosynthesis. This technology aims to make crops more efficient in producing the characteristics in which scientists wanted to achieve. The next on the list is the term chloroplast biotechnology, which is defined by Cruz et al. In the year 2021 as well as the process of and an advancement in the field of biotechnology where chloroplasts of certain plant species undergo alteration and sequencing, which aims to produce a more or highly efficient and resistant crops without harming the crops itself. What we must know between these two terms is that plastid transformation is the general term in the process of altering the DNA. Specifically, the ptDNA or pure plastid DNA results to crops efficiency, while chloroplast biotechnology is a specific one, which only involves the alteration of the chloroplast of the plant cell. But we must know that in this presentation, we are going to focus more about plastid transformation. The next slide contains a video in which the process of plastid transformation is included to further our knowledge of how plastid transformation works and the results of it.
So let's have a look at plastid transformation. So your basic requirement is a method of delivery, and by and large people have used biolistics. It is possible to use protoplasts, but the bialistic method works well and is easier to work with than protoplasts. And since the initial successful transformation in 1988 with Chlamydomonas, it's been repeated in a large number of other species, but it's still on the routine for tobacco. Now, biolistics will get your gene into the cell and into the chloroplast, so this is great. So virtually all your protocols are going to use biolistics. Some will use protoplasts, but most will use biolistics. Once your gene is in, then you need a good selectable marker in order to be able to select for your particular gene. The initial studies use the RRN16 gene, which is a 16S ribosomal RNA gene in the chloroplast. And you can get, this gene is susceptible to spectinomycin. So if you add spectinomycin to wild type cells, they will actually die off because it inhibits this 16S ribosomal RNA. However, a point mutation will actually confer resistance. So it's possible just to put in a single piece of DNA with this point mutation in it. It will go in by homologous recombination and you end up with a spectinomycin resistant variant. So the initial studies on plastid transformation use this RRN16 gene. Now, it's not a very effective way of actually producing resistance. The first reason for this is because it only actually acts on the actual site of action. So it's not doing anything about detoxifying the spectinomycin. It's just acting at the site of action on the 16S ribosomal RNA subunit. The other problem is you get lots of escapes. So the mutation rate and the translocation rate in chloroplasts is absolutely phenomenal. And what happens is that you end up with either the 16S ribosomal RNA point mutation actually reverting back, or much more commonly, you end up with false positives because of random mutations in the ribosomal RNA RRN16 gene. Far better is actually to put in some kind of detoxification method for spectinomycin. And this detoxification method is through this AADA gene, which is the 3-adenyl transferase gene. So you'll see that most protocols for plastid transformation nowadays will put in the AADA gene as a selectable marker. So this will actually adenylate spectinomycin and therefore make the spectinomycin inactive. And you can see here the construct of the gene. So the construct has got the -35 to -10 region, which is a promoter region, and also a region where you find the Shine-Delgado-like ribosomal RNA binding site. It'll also have a 5' untranslated region. These are regulatory elements, and also a 3' untranslated region. These regulatory elements that can actually help the expression of the ribosomal RNA. So you can get 100,000-fold variation in expression depending on these 5' and 3' untranslated regions. So you'll see that when they're developing and designing their plasmid, not only do you have to worry about which particular selectable gene you're going to put in, but you also need to worry about these untranslated regions, the 5' and 3' untranslated regions. So they tend to go for things like the PSBA gene 5' untranslated region, the TPSBA 3' untranslated region. And you can see they've got in there the AADA open reading frame and a Shine-Delgado sequence for ribosome binding. And you'll also need some sequences for homologous recombination. So outside of this particular vector that you're designing, you need to select a left and right hand side targeting region to allow the gene to go in by homologous recombination. Okay, and this neatly describes how the gene goes in. So you've got two genes and an intergenic region there, and the flanking regions are homologous to this intergenic region. And you've put in there two genes, the AADA gene is a selectable marker, the Cry2A AA2 gene, which is for Bacillus thuringiensis crystal toxin. Now, when you're looking at your promoter, it's important to have a very strongly expressed promoter in order to get expression. And some of these promoters are very highly expressed in green tissues, but they may not be expressed well in non-green tissues. So you have to look at where do you want this gene to actually be expressed. So there's a question mark there, not expressed in fruits. So some of these genes may not be expressed in, for example, etioplasts, which are not green, or in amyloplasts, which are not green. This is just a table to have a look at expression levels. So you can see, this is the crystal toxin, single gene expression in A for nuclear transformation. And you can see that around about 0.35% of total soluble leaf protein is this Cry2A A2 Bacillus thuringiensis crystal toxin. If you compare that to expression in plastids, we've specifically targeted it to chloroplasts, you can see that in leaves you're getting almost 50% of total soluble leaf protein expressed as Cry2 Bacillus thuringiensis crystal toxin. So it is possible to produce phenomenally high levels of expression in transgenic, transplastomic plants. One of the things that you have to do when you're selecting, you need a very rigorous selection for your transplastomic plants. The reason for this is that any one cell will contain numerous copies, numerous chloroplasts, so you may have 60 or 100 chloroplasts per cell. Each chloroplast will contain multiple copies of plastid DNA. So you can see here at the top left, you've bombarded your cell and you've got the plastid DNA into the cell. But the cell contains more than 10,000 plastid genomes per cell, because of the large numbers of chloroplasts, the large numbers of plastid DNA per chloroplast. So you get this into a single chloroplast. You then need to start selecting. So initially, you'll start to get overexpression of your gene when you're selecting for it. And you can see here, you've actually reduced the number of chloroplasts, so we're going through proplastids instead. So instead of one chloroplast that's got some transgenic DNA in it, in 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 chloroplasts, we've reduced the number of chloroplasts simply by going through proplastids rather than chloroplasts. So we're selecting one chloroplast out of 4 chloroplasts rather than 1 out of 10. And then you carry on with your selection and you finally end up with the gene stabilised. After several cycles of antibiotic selection, you end up with homoplasmy rather than heteroplasmy that we have here. And this is an example of selection of transplastomic clones by spectinomycin resistance. So we bombarded a whole load of leaf strips and we're getting the gene expressed. You can see here all of these leaf strips they're yellowed because of spectinomycin affecting the 16S ribosomal RNA, whereas one small cluster of cells has actually escaped the spectinomycin because it's been genetically transformed and it's regenerating into a plantlet. And then when we plate these out after a number of generations of selection, this is the transgenic tissue, this is wild type tissue. So to finish up, if we do a comparison of the nuclear and plastid genomes of angiosperms, a nuclear genome, 2 copies of each, many chromosomes, you could end up with thousands of genes per chromosome. But importantly, the arrangement of transcription of genes, each gene separate, individually transcribed, whereas you can have operons with plastid DNA.
The video discussed that plastid transformation uses biolistic method and thus produces crop efficiency throughout the process. To further our understanding of how this works and the results of it on the crops, we have to proceed first to the 4th slide, which will be discussed to us by Ms. Maria Irene Havinas.
The study's objectives or goals are to produce a new variety of high protein efficiency crops that can meet the predicted protein demand in year 2050, wherein presumpted to have a population of 9 billion. Furthermore, a crop that is not harmful to crops or consumers' health can reduce the impact of global warming and other environmental factors on crop production levels, as well as prevent and control the unpredicted mixing of crop characteristic or genes from wild relatives, ensuring that crop mutation is never a problem. If this study's goal can be achieved, it will benefit agriculture, pharmaceuticals, and other industries that use plastid transformation and chloroplast biotechnology.
Thank you, Irene. Knowing the objectives of the research or study makes it easier for us to understand what this study is for. Moving on the next slide, which contains the first topic of the article and the results of the research. This will be discussed to us by Mr. Julius Simbre.
And this is the result of the study. Plastid transformation is found to be one of the easiest producing products. Development of transplastomic plants, which includes different crops to produce new varieties, which contains high level of vitamins, antibodies, and other essentials contained by the crops. So the importance of transplastomic plants is very high expression levels of many pharmaceutically relevant proteins and ability to plastid to fold and assemble fully functional to proteins of various classes. So this is the example: Nicotiana tabacumbar, Pitted habana, Nicotiana benthamiana, and Nicotiana sylvestris. These are all is relatives to tobacco plants.
Plastid transformation is truly such an amazing process of how to make efficient crops, like the given example, the tobacco plant. By the way, thank you, Mr. Julius. Proceeding to the 6th slide, which contains some of the examples of plants which underwent plastid transformation. This will be discussed to us by Ms. Hannah May Bautista.
Hi, my name is Hannah May Bautista. So based from the plants that stated among the rest of the table is a plant that carries a higher possibility of stable transformation. But among the plants from the table, there is one species that is most likely the model, which is the tobacco plant. So why tobacco? Because tobacco obtains up to 100-fold frequency of transformation, so hindi lang siya for cigarette use. Tobacco still have a good benefit in the other sources like vaccine to treat illnesses. So whether tobacco is claimed to be an antiseptic, sedative, emetic, purgative, and useful in relieving pain. But how about the other plants like Arabidopsis thaliana, which is from the family of Brassicaceae, which also has a special case? That is because either Arabidopsis produces a nuclear homomeric acetyl CoA carboxylase or ACC2 gene, which is a form of fatty acid. But when plasmid translation is blocked by spectinomycin used as a selective agent in transformed tissues, the nuclear oxidase enzymes enables a limited fatty acid biosynthesis in plastids, permitting the rampant growth of untransformed callus material, which prevents the efficient obtaining of transformed plants. So we can tell that the transformation of Arabidopsis is not like the stability or stable transforming of tobacco. And when it comes to their genes, Arabidopsis have only 128 genes, while tobacco has 93,000 genes. So that is why tobacco is called the model of chloroplast transformation. And when we talk to the selection marker gene, which focuses on genetic transformation that allows plant cells to grow under condition that prevents the growth of untransformed tissue, it says that the experience of the plant is shown that 2 or 3 selection rounds have been sufficient to obtain homoplasmic plants. So that's all that I can explain. Thank you for listening.
That's a good analysis about the topic and why tobacco is a great example of crops which could undergo plastic transformation to efficiently produce essential properties for biopharmaceutical products. Again, thank you, Ms. Haname. And to further that, Ms. Angelica Estorco will provide us more knowledge of how important plastic transformation is to the production of crops which are essential to biopharmaceutical industry.
Chloroplast engineering for the improvement of biopharmaceutical industry. The goal of the study is to produce crops that can help the professionals to produce pharmaceutical products like medicines and vaccines is successful through the help of chloroplast engineering that made the crops highly efficient in producing the needed level of proteins and essentials materials needed to formulate pharmaceutical products. The article aims to produce medicines, vaccines, and other pharmaceutical products that has highly efficient level of proteins using crops like tobacco, corn, tomato, and other kinds of crops. The plan is enhancing the proteins and characteristic of the plant that is very important to biopharmaceutical industry. According to Rosales et al. And Morgenfeld et al., there have been significant results in choosing and promoting specific genes in crops to develop crops that will ensure efficient pharmaceutical goods like vaccines. The table shows the plastid transformations advances in biopharmaceutical products like human serum albumin is the product and the crop that is used is tobacco, and its total soluble protein or TSP is 11.1% and so on. The table are too long to be discussed, but that is one of the data that they gathered. The only one approved by the US Food and Drug Administration has been the human glucocerebrosidase made in carrot cell to treat Gaucher's disease using a nuclear expression system. So what is Gaucher disease? Gaucher disease is an inherited genetic disorder. It causes bone pain, anemia, enlarged organs, swollen, painful belly, and bleeding problems.
Thank you, Ms. Estorco. That's nice to know that plastic transformation is helping us solve health-related illnesses. And now to look closely of how it was formulated, a sample of process of how crops which contains essential materials to form medicines and vaccines will be discussed to us by Ms. Irene.
In making vaccines and medicines, there's a process that must be done to accomplish and formulate a good and effective vaccines and medicines. Crop with essential properties will be processed to formulate either a vaccine-based or oral-based medicine to counter or heal illnesses, making plastic transformation a very important part of biopharmaceutical industry.
That's incredible. Thank you, Ms. Irene. Let us now proceed to slide number 9, which will be discussed to us by Mr. Jolo Ordoña.
Chloroplast engineering to improve crop resistance and efficiency. According to Wu et al., development of durable and resistant crops from pests such as cryproteins which protect crops, specifically poplar crops, from insects, like Anticarsia gematalis, and the other because of the toxicity obtained by the plant from chloroplast alteration. Using a cry protein which acts like a pesticide or insecticide that can protect crops from those insects to make it resistant from pests. According to McBride et al. In the year 1995, this is a basis from the study made in 1995 where Cry1AC, a type of toxic protein, was developed to combat insects or pests. Cry1AC protoxin is a crystal protein produced by the gram-positive bacterium Bacillus thuringiensis. During sporulation, Cry1AC is one of the delta in the toxin produced by this bacterium which acts as insecticide. This also includes biotic and abiotic stress of the crops. Biotic factors are the living organisms in an ecosystem, and an abiotic factor is a non-living part of an ecosystem that shapes its environment just like water and an atmosphere, which both affects the crops, and those one of the concerns of professionals in the field deal with for the crops to be more efficient and protected. In the table 3, one of the examples is that the tobacco plant which contains Cry2AA2 to combat pests and insects to increase its durability and resistance.
Thank you, Jolo. To further analyze how cry proteins work, let us hear more of how it works from Angelica Estorco.
Example process of how cryproteins work. Cryprotein starts to work when an insect or pest consumes either end of the parts of the crops containing it. Upon consumption, the insect or pest will digest it, and the cryprotein will act as a poison which will surely kill the insect or pest eventually.
From a simple type of protein becoming an amazing and effective insecticides for crops, biotechnology is truly amazing beyond our comprehension. Thank you, Angelica. The next slide of this presentation will be discussed to us by Ms. Jenna Manalo, which contains information of why monocot plants can undergo plastid transformation.
Chloroplast engineering in monocots to improve protein production. The process of plastid transformation in monocot plants is still a big challenge to professionals in the field of biotechnology. And when the discovery on how to make monocot plants undergo plastid transformation, this will be a big achievement to the professionals as monocot plants are very essential in agriculture and even with biopharmaceutical industry. Monocot plants includes rice, maize or corn, and wheat, which are shown below.
Thank you, Ms. Jenna. There are truly still a lot of challenges on plastid transformation and chloroplast biotechnology. I'm sure that it will not take a lot of time for scientists to formulate a method to make monocot plants undergo plastid transformation. The last topic of the article, which is the pleiotropic effects experienced by the crops or plants which underwent plastid transformation, will be presented by yours truly, Christopher Chio. All types of experiments has its two sides, the bad and good sides. That's why science is said to be a double-edged sword. In the process of plastid transformation, the possibility of having plants or crops with abnormalities on their phenotypes has been one of the great concerns of the scientists and the researchers themselves. As alteration happens on the DNA of the crops, then too much changes as well as sudden or gradual changes within the DNA will cause undesirable effects on the plant as the plant relies on its cells to function well. In this next slide, examples of phenotypes or pleiotropic effects caused by plastid transformation on the tobacco plants is included, like the yellowing of the leaves, reduce in growth of the tobacco, and even sterility or inability of the male flowers of the plant to fertile the female flowers of the plant to continue the cycle, to pass down the original copies or characteristics of that specific variety. We're down to the second to the last part of the presentation, which contains the problems and challenges encountered by the study and the soon-to-be-solved mysteries of the study. This will be discussed to us by Miss Irene and Hannah, together with Mr. Julius and Jolo.
Plants who underwent the process of plastid transformation thus result on abnormal phenotypes and mutation because throughout the process it implemented to insert new DNA and deleting traits of the plants, and also transformed plants can hybridize with wild relatives allowing transgene to be dispersed and controlled, which commonly known as crop mutation.
And unsolved and is still undergoing discovery of how to make monocots undergo plastid transformation to solve the possible lack of protein supply in the future for human consumptions. So, as plastid transformation grown, I became more and more a challenge. It has been a big problem in biotechnology field. Many undergoes experiments and research to discover a possibility of monocots undergoing plastid transformation is yet to discover, but this might cause some challenge.
The limitation of crop resistance over herbicides and pesticides. So there are still some cases that a crop has the limitation to resist herbicide tolerance and pesticide resistance. For example, when you are using herbicide, which is the ability of plant to survive and reproduce to a dose of herbicide normally lethal to the wild type. So if you are using a type of herbicide, there is a possibility that the plant will not survive. But in other cases, it helps the plant to eliminate unwanted plant-like weeds. Otherwise, pesticide also have some cases that the pest can tolerate the resistance of a pesticide. So some of the solution of this is the cryptophyll protein, which protects the plant from insect or weeds. But sometimes, the problem is insect can also develop resistance to cytoplasmic cryptophyll. So between plants and pesticide, there is a limitation between both parties to resist or tolerate some circumstances to pesticides and herbicides. So the only solution for this is to improve the herbicide resistance and pesticide to cope up with the thing we call abiotic and biotic stress tolerance, which controls a plant by tolerating the temperature, weather, insect, and other risks to plants.
Negative views and feedback from public over chloroplast biotechnology and plastid transformation, which might cause health-related issues both in plants and humans. So in this case, they need to assure that chloroplast biotechnology and plastid transformation can't have a bad effect and can't be harmful from the humans.
This study truly still holds a lot of undiscovered facts behind the processes of plastid transformation. And now, as we end this presentation, a summary is included by our group that I will read for us to come up with conclusions and answers to the given questions. The development of crop efficiency with the use of chloroplast biotechnology and plastid transformation gave rise to the improvement of different fields that concerns it, such as agriculture and pharmaceutical industry. The study aimed to introduce the importance of improving crops through genetic or DNA alteration to produce the wanted effects, specifically producing proteins to meet and supply the demand, which was seen to increase by the year 2050. The study was found to be successful in developing new technologies and ways or methods to increase crop efficiency in terms of resistance to environmental factors and others which affects the crops and increase productivity with the use of biotechnology. Despite some difficulties encountered and mysteries yet to be solved, the study unveiled the secret and build or establish a new stage of improvement in the field of biotechnology. Answering the questions. The first question is to be answered by Miss Irene.
The process used by the study to further the understanding of how plastid transformation is called bioelastic method. Bioelastic is a high-speed particle bombardment approach for delivering nucleic acid to cells. To transfect cells or organelles, the approach uses nucleic acid-coated particles pushed by a pressurized pistol or gene gun.
Thank you, Miss Irene. Second question will be answered by me, yours truly, Christopher Gio. If plastid transformation will be soon available to all, recommending it to farmers and some of agriculturists who doesn't know about it will never be a bad idea. Plastid transformation, as discussed, aims to produce more efficient crops when it comes to different aspects like protein production and other essential materials for biopharmaceutical industry. Applying this technology to solve common problems and major problems of agriculture like growing or increasing demand for food supply, plastic transformation will be a win-win solution. Making the production cost cheaper and time to produce these products shorter will surely change the current situation of our world, where there's a big problem to global food security. And now, the last question will be answered to us by Miss Irene.
The significance of the article is how useful plastic transformation is on our time. We all know that plastic transformation aims to equip different industries like agriculture and biopharmaceuticals with the capabilities and materials to meet and supply the demand of certain types of products or goods needed by the ever-increasing population of our world. When it comes to limitation of the article, the discussion of how to make monocoque plants undergo plastic transformation and the further understanding of the side effects of plastic transformation on both plants and humans which consumes them to create a limited content or discussion of how effective and important plastic transformation is.
Thank you, Miss Irene. That's it for our presentation. Again, we are Group 4 of BSA 1C, proudly presented to you Article 4 titled Plastic Transformation, Advances and Challenges for its Implementation in Agricultural Crops. Thank you for listening, ma'am, and keep safe always.